Thanks, Demir. It's really been fun to participate in this workshop. And I have this topic here is a little bit off course, but I hope you find it intriguing. We certainly do. So about 15 years ago, we had a collaboration with Yuri Vasiliev, who was a prominent Russian cell biologist. He's quite old now. And what he and Olga Petushkina realized was that when they allowed cells to spread on a substrate and depolymerize their microtubules, they run through a very regular oscillation with a period of about 1 minute. And I felt almost from the beginning that this was a good opportunity to meld theory and experiment. And so you might ask why we would be interested in this. Well, one is I think it gives us a window into interactions of the actomyosin and microtubule subsystems on a global scale. It's the opportunity to study the cell cortex, which is largely an undefined entity. It has some physiological relevance. This is probably a bit far-fetched, oscillations in cardiomyocytes, but there are oscillations in the contractile ring of dividing cells. And also, we feel it might be a model, tractable model for a certain type of amoeboid movement. And in general, it's a prototype for modeling complex mechanochemical phenomena on the cell scale. So what I'd like to discuss is the experiments, then a little bit on the interplay of theory and experiments, and then a little bit on the relevance to amoeboid movement at the end. So the people who have worked on this are both theoreticians, Tim Elston and Greg Forrest at UNC, Chi Wang and Xiaofan Yang at University of South Carolina. Then the experimental work has been done predominantly by Marina Kapustina and also some by Nancy Castigliola when she was a student in biophysics at our school. So in terms of the current and past theoretical thrusts, and experimental thrusts, if we look at this phenomenon, first of all, the regular period is easy. It forms a sort of a assay parameter for biochemistry and molecular biology, so that we can take the oscillating cell and plot the movement of center of mass of a phase image as a function of time. And then we can do a Fourier transform, and from the normalized power spectrum, we can pull out the dominant periods of this movement. The first thing we realized was that microtubule depolymerization increases the amplitude of oscillations. So if we look here at a cell in which here we're expressing a fluorescent protein tubulin, we can see that the oscillations are not that big in amplitude as detected here. But if we take colcimid and we incubate with colcimid, you can see that the oscillations grow massively in amplitude. So we knew so microtubules are a way to enhance this phenomenon, although it occurs spontaneously as well. Now, what we know is during this phenomena, fixed cell studies tell us that the actin cortex, and we're standing with phalloidin here, is highly polarized. This is probably the contractile portion. And we know that latrunculin does not inhibit this phenomenon. We know that if we stain for phosphomyosin 2, we can see that this would be the contractile portion of the cortex. And as we were talking about yesterday, you can see that, at least in fixed cells, when this side of the cell squeezes, the nucleus is pushed way to the other side and highly distorted. And we have three-dimensional renditions of the phalloidin stain and the nuclear stain here, and the myosin, active myosin stain here, and the nuclear stain. So if we look at this in wide field microscopy, this is the DIC image. And this is now the image of the cortex using GFP LifeAct, which binds, side binds to actin filaments. Then we can see the dynamics of the phenomenon. This is now in wide field microscopy, but I'll show you more in confocal in just a second, which makes it a little clearer. Here we have another wide field. This is the LifeAct outlining the cortex, and the myosin is in orange here. This is just an expressed myosin light chain, not detecting myosin activity, but localization. And if we look at this, we can see that the phenomenon in more detail with the cortex here, and you can begin to see evidence of a traveling wave. And that's what we'll discuss more as we go on. Now, we can visualize this in 4 dimensions, that is, taking rapid slices in a confocal microscope, slowing down the phenomenon. And when we look at, and we're now looking at the cortex again with LifeAct, and this is a 3-dimensional reconstruction as a function of time, you can see this, what we call a periodic protrusive phenotype here, where the protrusion is coming out here. Now, as I said, the nucleus, here we have a nucleus in living cells that are oscillating, that is stained with Herc stain, which stains the DNA in nucleus. And you can see that the nucleus is really pushed about the cell with great force, apparently. And you can see it's becomes highly distorted. And that gets to a point we were discussing yesterday about how the nucleus is distorted in transendothelial migration. Now, as I said, there are cortical actin traveling waves. So here what we've done is to express a plasma membrane tracker, which is a RFP that's dually acylated and plugs into the inner leaflet of the plasma membrane. That's in red, and the cortex now is in LifeAct. And you can watch the traveling wave. This is a bit like a biological Rubik's Cube to me. It's hard to intuit, but the traveling wave is going to be moving this way. So here you see the wave moving around the cell, and you can't look at this too long without going crazy, but but it does, it indicates on the mesoscale how deformable the cortex is. Now, the reason that the red stain is broadened more than you would expect for a membrane stain is that what's happening apparently is that the cortex and membrane are getting progressively folded until we get big folds here. And if you look, you can show that by fixing the cells that are oscillating and going through a thin section transmission electron microscopy. And what you see is in the protruding part, it's quite smooth. But in the folded part, you can see evidence on the ultrastructural scale of these folds. And these little vesicles are probably folds that are coming up in section. So you see they look like vesicles. And if you took a control cell, like a CHO cell here, that's spread on the substrate, there's no such evidence of that kind of folding. Now, we attempted to estimate carefully the degree of compaction and of the folded membrane cortex, and it varies between 2 and 6 from both. That's in agreement with both the TEM and the fluorescence microscopy. So a question comes up. Is this classical membrane blebbing? So in membrane blebbing, what you have is the cell here. There's tension in the cell and in the cortex. And stochastically, somehow the cortex breaks. And then cytosolic pressure moves a pure membrane bleb out here like this. And then with time, cortex reappears and stabilizes that bleb and actually retracts it back into the cell and to begin again when another break in the cortex appears. This apparently is not what's happening in our case. So here what Marina has done is look at an oscillating cell, and we're going to take a line scans across this region of both membrane and cortex. And what we see is here is the membrane stain, and in blue is the cortical stain. And you can see even in the protruding part, there is always a membrane accompanying cortex. And when the protrusion is on the other side of the cell, the same thing obtains. In the thick part, you see the membrane stain here and the cortical stain. But in the protruding part, you see membrane and cortex coincident, or nearly coincident. And you can plot the intensity of membrane versus intensity of the cortical actin, and you can see it's a linear relationship. So we never have it doesn't appear that we ever have a situation where the cell blebs without cortex under it. So that led to a hypothesis that compression and dilation of the membrane-cortex couple generates rapid changes in cell shape. And you can go the other way. So what Marina did was to take a spread cell here and rapidly detach it with trypsin. Now, in order to accommodate all the surface area in this small sphere, you'd have to have a degree of compaction, roughly 6, to fold up the membrane cortex. And that sets the cell into oscillation somehow. And it's not clear why, but that instability sets the cell into oscillations. Now, what about the regulatory aspects of this periodic protrusive phenotype? And I'm not going to go into this in detail because of time. But we know now that calcium is not involved. We originally thought calcium was the driver. At least one of the major drivers is Rho-A, and also phosphoinositides are involved. So Rho-A activation is necessary but not sufficient to induce oscillations. So we know that oscillations are inhibited by Rho-A siRNA, as Ann explained yesterday. And downstream, there's a downstream effector called Rho kinase, and if we inhibit that, we stop the oscillations immediately. But the oscillations are not enhanced by global lysophosphatidic acid or thrombin. That's important because those are generalized activators of Rho-A. And that means there must be a spatial temporal aspect to Rho activation. And indeed, if you use a positional biosensor, which is 3 times GFP hooked to an Rho binding domain, that will detect active Rho. Okay? And this is a very short movie, but you will see that the active Rho, the Rho is at least accumulated in the folded regions. Whether it's increased in density, we can't say because we haven't done that comparison. But certainly there's active Rho in the region of heavy folding. Now, the phosphoinositides, which are these lipids which are on the inner leaflet of the plasma membrane and are often involved in regulating actin polymerization, they're also involved. So it's phosphoinositide, PIP phosphate, PIP2, and PIP3. We know that because if we inhibitors of phospholipase C inhibit oscillations. And phospholipase C converts PIP2 to IP3 plus diacylglycerol. Okay? Also, inhibitors of PI3 kinase, Wortmannin and another one I'm forgetting, block oscillations. And what that does is you take PIP2 and the kinase, PI3 kinase converts PIP2 to PIP3, where we have 3 phosphates on the inositol head group of the phospholipid. And indeed, if you take another positional biosensor, AKT pH domain, tagged to EGFP, you will see that it tracks, this is going to recognize PIP3 in the inner leaflet. You will see it tracks this traveling wave. And you'll also see extensive cytoplasmic flows in this, in this movie. So another thing theory has to predict eventually is these immense cytoplasmic flows in this phenomenon. Now, how do we explain a traveling wave? These are some general features, and I am not a traveling wave theorist, so forgive me. But one of the things is that we have to break symmetry to start the wave. And usually what's invoked is a Turing instability to amplify stochastic fluctuations. And then to get a wave to travel with a front edge and back edge, we require a local negative feedback with a delay. So if this is the direction of the traveling wave, there is a negative feedback that helps to define the trailing edge of the wave. Well, we can't say much about the details of this, but what it might mean is that we might have a positive feedback or even a global positive feedback that begins to, say, concentrate rho in this region, maybe due to a GEF binding to PIP2 in the inner leaflet. And the positive feedback continues, and we get initiation of a negative feedback. And in this region, a negative feedback predominates. There's probably also a signal that releases these major folds. And then we now have an excess of sort of untethered membrane cortex couple. And the cytosolic pressure then inflates that excess, producing a rounded protrusion. That would be one way of looking at it. So how can we go about modeling this periodic protrusive phenotype? The general plan, and I'm not in the theoretical end of this, but is to take a spatially localized ODE, PDE description of Rho A regulation, couple that active Rho A concentration to cortical dynamics, and do a multi-phase active gel model of the cortex with a forcing function that's provided by the Rho A regulation, the active Rho A. Well, in brief, a multi-phase cell model is part of this phase field modeling that Chi Wang and Greg Forrest are actively engaged in to model interfacial phenomena. I'll give you an example in just a second. But in general, what we have is a surrounding medium. We have the membrane as a closed elastic surface. The cortex is going to be modeled as an active pneumatic phase. Joanny in Paris has also done this. And then the cytosol will contain the regulators that regulate the mechanism that propagates the traveling wave. And this sort of phase field modeling, this is an example of where it's been used. If you start with a pneumatic phase, a pneumatic liquid crystal as a sphere, and you place it in a flow field, then what you can predict is the distortion of the pneumatic phase and also the flows that exist within that phase. So an early attempt at this was to take a pneumatic phase and circulate a contractile region that's totally artificial, but you begin to see some similarities to the experimental phenotype. The reason it's totally artificial is the forcing is done with exactly the angular velocity that the wave experimental wave is running at. But that is the general idea, and also to see if this multi-phase model can predict the internal flows. Well, finally, are these oscillating models cells a model for pure Blebby type amoeboid locomotion. And amoeboid locomotion is seen in various forms in leukocytes, dictyostelium, and metastatic cancer cells. And it is not really studied nearly as much as is mesenchymal locomotion. So Peter Friedel has made this classification, which we'll see if stands the test of time. But he defines an amoeboid Blebby form of locomotion in a three-dimensional matrix as actually not grabbing onto, via integrins or something, onto the matrix filaments, but propelling itself through interstices in the matrix, mainly by cytosolic pressure. And then there's an amoeboid pseudopodial, filopodial type that actually does extend lamella and grab onto filaments, some sort of hybrid of these two mechanisms. And then there's the mesenchymal or fibroblastic form where the cells are actually tracking along via integrin and other adhesions along filaments in the extracellular matrix. Well, are our cells relevant to amoeboid movement? That's an open question. This is a famous movie, I'm sure many of you have seen, of a leukocyte chasing, PMN chasing a bacterium here by Dave Rogers. It's kind of pixelated. This is in a field of red cells, and you can see eventually it will get this bacterium. And this is a three-dimensional view of one of our cells oscillating. And at least you can see some superficial similarities. Now, the similarities actually they're qualitative, but there are a number of them that might be pointed out. First of all, in amoeboid movement, usually adhesion is weak, and that's true in the periodic protruding cells. There's a lack of obvious polarity except when you have a chemotactic gradient. That's true in both of these cells. Both have rounded protrusions. Both presumably do not do extracellular matrix proteolysis, but as we heard from Ann, that's probably, in the case of amoeboid movement, may well be disputed. Both usually require high Rho A activity, but low RAC activity, and both involve phosphoinositides. So there's some, at least superficial, similarities between our phenotype and a Blebby type of amoeboid movement. Well, do these cells actually move? If you get them on a substrate of proper adhesivity, they will. I mean, if you have too strong adhesivity, they won't move, or too weak adhesivity, they won't move. But it turns out that oscillating CHO cells on collagen without depolymerized microtubules, spontaneously oscillating, and this shows the traveling wave moving around, these cells will move, and they'll move at velocities of something like 0.5 to 5 microns a minute. Also, we can take the migration of the mammary breast carcinoma cells, and we can put them on serum-coated glass, and they will undergo this periodic protrusive phenotype whilst migrating. So there is evidence that this may be a model for a certain type of amoeboid movement that's even theoretically tractable. And finally, here's some very data in the last couple of weeks where Ramiro Cucciero has put these cells in a three-dimensional collagen matrix. So these cells are deep within this three-dimensional collagen matrix. And what if we watch them, particularly watch the cells up here, these were embedded in the matrix, they were oscillating, and you can see that they can undergo some type of migration, particularly up here. There's a situation where they will almost look like a sort of a leukocyte moving through the matrix. So these are open questions and, as to the relevance of this phenotype, but we hope it's relevant. Oh dear. I forgot to turn my phone off. So in summary then, spreading cells or rounded cells will undergo morphological oscillations that are enhanced by microtubule depolymerization. The cortex is highly polarized in protruding cells, and dense regions of the cortex move as a traveling wave. And this implies a compression-dilation model of membrane-cortex couple. RhoA and phosphonosides are key regulators. Calcium presumably has a lesser role. Multiphase modeling shows promise in understanding this phenotype on a theoretical basis. And periodically protruding cells can migrate given the appropriate substrate adhesivity. So this is possibly a tractable model for amoeboid migration in 3 dimensions. Time will tell. So I'll stop there and take any questions.